Part 2. Bismillahirrahmanirrahim. This is another online class. Our topic today is reproduction as a function of cell. Under reproduction as a function of cell, the main thing to discuss is meiosis. I told you in the last discussion that we have two types of cell division based on the behavior of chromosomes. One of these division is meiosis, which is reduction division. This meiosis is a nuclear or cell division in which the daughter nuclei contain half number of the chromosome and features as the parent nuclei. That is, meiosis is a type of cell division in which a diploid cell undergoes two successive divisions. The meiosis is subdivided into two divisions. Namely, we have meiosis I and meiosis II, or the first meiotic cell division and the second meiotic cell division. Then, what we are going to really discuss in this online class is the meiosis I, or the first meiotic cell division. We have 4 stages under meiosis I. The first one is prophase stage, the second one is metaphase stage, the third one is anaphase stage, while the last one is telophase stage. The prophase stage, which is the first stage, that is prophase I, is subdivided into 3. The first one is early prophase I, the middle prophase I, and the last one is the late prophase I. In the early prophase I, chromosomes become visible as long threads. That is, the chromosome become elongated and form a network of fine threads called chromatid or chromatin threads. Then, this is chromatin threads. These are the chromatin threads. Then, in the prophase I, in the early prophase I, you can see that the nuclear membrane is visible. The nucleus also is visible. Then the central form esters. Central form esters. Then let us look at the middle prophase I. In the middle prophase I, homologous or matching chromosomes become thicker and shorter and clearly seen. Homologous chromosome is the two sets of chromosomes in a diploid cell. Homologous or bivalent chromosome is the two sets of chromosomes in a diploid cell. Spindle also is formed at the middle prophase. Spindle, that is, spindle fibers are formed. So these are the homologous chromosomes that become visible. These are the spindles that are formed. Still, division of cell cannot take place because the nuclear membrane is still visible in the middle prophase I. Then the late prophase I, you can see that the nuclear membrane will start disappearing in the late prophase I. In the late prophase I, each chromosome reproduces replica of itself. That is, it becomes double-stranded. Double-stranded. It becomes double-stranded. They replicate. This is chromosome. This is chromosome that replicates that reproduces replicates of itself. This is another one. This another one. It reproduces replicate of itself. Double-stranded. Now, the nuclear membrane starts disappearing. Look at the dotted line. This dotted line represents nuclear membrane. It starts disappearing. As nuclear membrane starts disappearing, the nucleus also starts disappearing. Then, cross-link called chiasma or cross-links called chiasmata. Chiasmata is plural, while chiasma is singular. Then cross-links called chiasmata are formed between homologous chromosomes. These chiasmata help to hold or link two homologous chromosomes together, and thereby enhances the exchange of genes or genetic materials between homologous chromosomes, which lead to variation or mutation. Variation is the observed natural differences, or is the natural observed differences between organisms of the same species. Mutation. Mutation is the sudden change in the number of chromosomes in the nucleus of a cell. Or you can say mutation is the sudden change in the arrangement of genes or DNA molecules in the nucleus of a cell. When you get to SS3, your teacher will discuss variation and mutation for you vividly. So this is chiasma that link homologous chromosome together, or hold homologous chromosome together. Chiasma. So you can see the link together here. Chiasma. It is chiasma that links them together. Now, look at genes. The genes, there is going to be exchange of genes between these chromosomes and these chromosomes. This chromosome and this chromosome, there is going to be exchange of genes between them. And it is the chiasmata or chiasma that enhances the exchange of genes between homologous chromosomes. Let us look at the metaphase 1. In the metaphase 1, the nuclear membrane disappears completely. The nuclear membrane disappears completely. We look at this one, there is no nuclear membrane. Then the nuclear membrane gets dissolved. That is, it is the same thing with this thing is the same thing with what I have just said, that nuclear membrane disappears completely. That is, nuclear membrane gets dissolved in the cytoplasm completely. Then bivalent chromosomes are arranged at the equator. Look at the bivalent chromosome. They are arranged at the equator and attached to the spindle by the centromere. This is centromere. This is another centromere. Let us look at the anaphase 1. In the anaphase 1, each member of the bivalent chromosomes moves apart and toward the post or end of the cell by the shortening of the spindle fibers. By the shortening of the spindle fiber. Look at the spindle fiber. The spindle fiber is shortening, and thereby the chromosomes, the homologous chromosome, move to the end of the pole for the division of cell to take place. Look at it. This is spindle fiber. It's shortening to enhance the movement of homologous chromosome or bivalent chromosome to the end of the cell for the division of cell to take place. Then let us look at the telophase stage. At the telophase stage, the bivalent chromosome arrive at the pole of the cell. A new nuclear membrane is formed. Look at the nuclear membrane. Immediately the bivalent chromosome reach the end of the cell will start dividing. Look at the cell. It will start dividing. Then bivalent chromosome arrive at the pole. Then a new nuclear membrane is formed around each group of chromosomes. Immediately the bivalent chromosome arrive at the pole, the nuclear membrane is formed. A new nuclear membrane is formed, and nucleus also will form. Then chromosome uncoil. The chromosome will become uncoiled. Now let us look at the differences and similarity between mitosis and meiosis. But before we discuss differences and similarity between mitosis and meiosis, I want you to take note of this thing, that this division of cells is the reduction division of meiosis. Then no cleavage of cytoplasm take place. That is, there is no crossing over in this first meiosis or first meiotic cell division. In the meiosis one, there is no crossing over. The short interface that follow does not witness replication of DNA as in the interface of mitosis. That is, the interval between the interface in the telophase 1 and prophase 2 is too short. Therefore, there is no replication of DNA as in the case of mitosis. The cell proceeds to the second division of mitosis. So we discuss the second division of meiosis in the next online class, inshallah. Now, differences between meiosis and mitosis. Now, if you look at mitosis, it takes place in the somatic cells. Mitosis takes place in the somatic cells, while meiosis takes place in the reproductive cells or sex cell or germ cells. So somatic cell is also referred to as body cells. Example of body cell is skin. 2. No pairing of chromosomes. There is no pairing of chromosome in the mitosis, whereas there is pairing of homologous chromosomes in the meiosis. In the mitosis also, only one nuclear division takes place. That is, one nuclear division occurs. But in the case of meiosis, 2 nuclear division takes place. Then there is short prophase stage in mitosis. In the case of meiosis, there is long prophase stage because we have early prophase 1, middle prophase 1, and late prophase 1 in the case of meiosis. But in the case of mitosis, we have only one prophase stage. Then no chiasmata in the mitosis. There is no chiasmata or chiasma in the mitosis. Then chiasma occurs in the meiosis. In the mitosis, there is no exchange of genes. That is, there is no exchange of genetic materials. But in the case of meiosis, there is exchange of genes or genetic materials. It is this, I told you that it is this chiasma that enhances the exchange of genetic material between 2 homologous chromosomes. Another one is that daughter cells in the mitosis contain the same number of chromosomes and features as the parent cell. But in the case of meiosis, daughter cell contain half the number of chromosome and features as the parent cell. I told you the reason for that at the beginning of this class, at the beginning of this teach. Now, so If you look at the daughter cells, in the case of mitosis, they are identical. Daughter cells are identical to the parent cell, but in the case of meiosis, variation occur in the daughter cells. In the case of mitosis, there is no crossing over, whereas there is crossing over in the case of meiosis. There is crossing over in the case of meiosis. This is crossing over. Crossing over. So this is crossing over. Now let us look at the similarity between the two. Similarity between mitosis and meiosis. Interphase is the same in mitosis and first meiotic cell division. Interphase is the same in the mitosis and the first meiosis cell division. If you look at the in Prophase 1, you will see nuclear membrane, mitosis, nucleus, nuclear membrane, nucleus, the elongated chromosome that form network of fine thread called chromatin. The same thing happens in the interphase stage in the mitosis. 2. Centrioles divide in both mitosis and meiosis at interphase. That is, doubling of chromosomes takes place in the interphase stage in both mitosis and meiosis. Number 3. Nuclear materials dissolve at prophase in both mitosis and meiosis. Number 4. Spindle is laid down in the cytoplasm by centrioles, which divide in interphase. Spindle is laid down in the cytoplasm by the centrioles, which divide in interphase. Both mitosis and meiosis form new cells from pre-existing cells. This is the end of this online class. And for your assignment, make sure that you check the assignment portal. Assalamu alaikum.